We will continue our discussion on molecular and cellular radiation biology. This presentation will review ionizing radiation effects on DNA, cell survival curves, and the law of Bragonian-Tribandau. There are several possible effects of ionizing radiation on DNA: a single strand break, double strand break, and double strand on same rung break. In a single strand break, also called a point lesion, occurs when ionizing radiation interacts with a DNA macromolecule and the energy transferred ruptures one of its chemical bonds and severs one of the sugar phosphate chain side rails. Such as a single alteration along the sequence of nitrogenous bases can result in a gene abnormality. Point lesions commonly occur with low LET radiation. Repair enzymes, however, are often capable of reversing this damage. Further exposure of the affected DNA macromolecule to ionizing radiation will likely lead to additional breaks in sugar phosphate molecular chains. These breaks may also be repaired, but double-strand breaks are not as easily repaired as single-strand breaks. If repair does not take place, further separation can occur in the DNA chains, threatening the life of the cell. Double-strand breaks happen more commonly with high LET exposure. When high LET radiation interacts with DNA molecules, the ionization interactions may be closely spaced that both strands of a DNA chain are broken. Should both strands be broken at the same nitrogenous base rung, the result will be the same as if both side rails of the latter were cut at the same step. The DNA ladder would be chopped into two pieces. This situation will result in the associated chromosome to be broken itself. Thus, some types of chromosomal damage that are specifically caused by high LET radiation are related to double-strand breaks of DNA. Because the chance of reversing this type of damage is very low, the possibility of lethal alteration of nitrogenous bases within the genetic sequence is far greater. As mentioned earlier, when two interactions, one on each of the two sugar phosphate chains, occur within the same rung of the DNA ladder-like configuration, the result is a broken chromosome, with each new portion containing an unequal amount of genetic material. If this damaged chromosome divides, each new daughter cell will receive an incorrect amount of genetic material. This will culminate in either death or impaired functioning of the new daughter cell. Interactions of ionizing radiation with DNA molecules may cause the loss of or change in a nitrogenous base of the DNA chain. The direct consequence of this damage is the alteration of the base sequence within the DNA molecule. Because the genetic information to be passed on to future generations is contained in the strict sequence of bases, the loss or change of a base in the DNA chain represents a mutation. It may not be reversible and may generate acute consequences for the cell, but more important, if the cell remains viable, incorrect genetic information will be transferred to one of the two daughter cells when the cells divide. Crosslinks are chemical unions created between atoms by the single sharing of one or more pairs of electrons. Crosslinks involving DNA are usually caused by high LET radiation. With low LET interactions, crosslinks are most likely caused by the process of indirect interaction. After irradiation, some molecules can fragment or change into small, spur-like molecules that become very interactive or sticky when they themselves are exposed to radiation. Such sticky molecules can facilitate cross-linking by attaching or connecting to other macromolecules or to other segments of the same macromolecule chain. Cross-linking can occur in many different patterns. Intrastrand cross-link occurs when cross-linking is formed between two places on the same DNA strand. Interstrand cross-link can occur between complementary DNA strands or between entirely different DNA molecules. When changes occur in the DNA molecule, the chromosome exhibits the variation. Because DNA modifications are discrete, they do not inevitably result in observable structural chromosome revisions. However, if these discrete effects are numerous enough, such as may be brought about by exposure to high LET radiation, then an observable structural chromosome alteration process is possible. One effect of ionizing radiation on chromosome is radiation-induced chromosome breaks. After irradiation and during cell division, some radiation-induced chromosome breaks may be viewed microscopically. These changes are manifested during the metaphase and anaphase of the cell division cycle, when the length of the chromosome is visible. Both somatic and genetic chromosomes are subject to radiation-induced chromosomal breaks. If radiation interacts with and deactivates the cell's DNA, cell death occurs. After chromosome breakage, two or more chromosomal splinters are produced. Each of these has a fractured extremity. These broken ends are chemically very active and therefore have a strong tendency to adhere to another similar sticky end. The broken fragments can rejoin in their original configuration. They can fail to rejoin and create an aberration, a lesion, or anomaly. Or they can join to other broken ends and thereby create new chromosomes that may not look structurally altered compared with chromosome before irradiation. Chromosome anomalies include chromosome aberrations, again those are lesions or anomalies, but also include chromatid aberrations. You can read more about these anomalies in your textbook. Ionizing radiation interacts randomly with matter, giving up energy in the process. Because of this energy transfer, exposure to radiation could lead to the occurrence of structural changes in biologic tissue. Some of the consequences to the cell from structural changes within the nucleus consist of restitution, deletion, broken end rearrangement, and broken end rearrangement without visible damage. The process of restitution is whereby the breaks rejoin in their original configuration with no visible damage. In this case, no injury to the cell occurs because the chromatid has been restored to the condition it was in before irradiation. The process of healing by restitution is believed to be the way in which 95% of single chromosome breaks mend. The process of deletion is one in which a part of the chromosome or chromatid is lost at the next cell division, thus creating an aberration known as an acentric fragment. This results in a cell mutation. Broken end rearrangement, whereby a grossly mishapen chromosome may be produced. Ring chromatids, dicentric chromosomes, and anaphase bridges are examples of such distorted chromosomes and chromatids. This also results in a cell mutation. Broken end rearrangement without visible damage to the chromatids is whereby the chromatid's genetic material has been rearranged even though the chromatid appears normal. Translocations are examples of such rearrangement. This results in a cell mutation. For the cell as a whole, damage to the cell's nucleus reveals itself in one of the following ways: instant death, reproductive death, apoptosis or programmed cell death, mitotic or genetic death, mitotic delay, or interference with function. Instant death of large number of cells occurs when a volume is irradiated with an X-ray or gamma ray dose of approximately 1,000 gray in tissue in a period of seconds or a few minutes. This large influx of energy causes gross disruption of cellular form and structure and severe changes in chemical machinery. As a result of receiving such a massive dose of ionizing radiation, the cell's DNA macromolecule breaks up and cellular proteins coagulate. Radiation doses high enough to cause this type of damage are vastly greater than those used for diagnostic examinations or even standard therapeutic treatments. Reproductive death generally results from exposure of cells to doses of ionizing radiation in the range of 1 to 10 gray in tissue. Although the cell does not die when the reproductive death occurs, it permanently loses its ability to procreate but continues to metabolize and to synthesize nucleic acids and proteins. The termination of the cell's reproductive abilities does, however, prevent the transmission of damage to future generations of cells. Apoptosis, or programmed cell death, is a non-mitotic or non-division form of cell death that occurs when cells die without attempting division during the interphase portion of the cell life cycle. Apoptosis occurs spontaneously in both normal tissue and in tumors. It can occur in human beings and other vertebrate animals and amphibians. An example of this process is tadpoles losing their tails. Certain types of programmed cell death are integral to the development and maintenance of organisms. Many types of cells are destined to die for the good of the organism. An example of this is skin cells. When cells die, they break up and are ingested by other cells. The more radiosensitive the tissue, the less exposure is required for apoptosis to occur. Ionizing radiation can adversely affect cell division. It may stunt the mitotic process or permanently inhibit it. Cell death follows permanent inhibition. Mitotic or genetic death occurs when a cell dies after one or more divisions. Even relatively small doses of radiation have a possibility of causing this type of cell death. The radiation dose required to produce mitotic death is less than the dose needed to produce apoptosis in slowly dividing cells or non-dividing cells. Exposing a cell to as little as 0.01 gray tissue of ionizing radiation just before it begins dividing can cause mitotic delay, the failure of the cell to start dividing on time. After this delay, the cell may resume its normal mitosis function. The survival curves for mammalian cells was constructed from a series of experiments. Cells were grown in a culture in a lab and then exposed to specific doses of radiation. The scientists measured the cell's ability to divide and survive. The experiments are repeated for several dose amounts. The cell survival curve shown on this slide is for the same cell line irradiated with both low and high linear energy transfer radiation. With low LET radiation, a shoulder to the curve at lower doses indicates the cell's ability to repair some damage. High LET radiation typically has no shoulder, thus indicating that little or no repair takes place. Cell radiosensitivity was discovered by Jean Bergoni and Louis Tribendau in 1906. After observing radiation effects on testicular germ cells of rabbits, they established that radiosensitivity was a function of the metabolic state of the cell receiving the exposure. Their findings became known as the law of Bergoni and Tribendau. It states that the radiosensitivity of cells is directly proportional to their reproductive activity and inversely proportional to their degree of differentiation. Thus, the most pronounced radiation effects occur in cells with the least maturity and specialization or differentiation, the greatest reproductive activity, and the longest mitotic phase. Although the law was originally applied to germ cells, it is actually true for all types of cells in the human body. Consequently, within the realm of diagnostic imaging, the embryo fetus, which contains a large number of immature, non-specialized cells, is much more susceptible to radiation damage than is an adult or even a child. Again, cell radiosensitivity is something all radiographers need to be aware. The amount of radiant energy is important in determining possible effects to the cell and tissue. LET affects radiosensitivity, with high LET causing more biologic damage. Since radiographic procedures use low doses of radiation that are low LET, very few cells are killed with this type of radiation. However, in radiotherapy, the presence of oxygen plays a significant role in radiosensitivity and thereby treatment efficacy. Sometimes therapies use a hyperbaric chamber to increase the effective treatment to a tumor. More free radicals are released in the presence of oxygen, making this a viable treatment option. We have discussed the more mature and specialized in performing functions a cell is, the less sensitive it is to radiation. However, ionizing radiation does affect human cells and tissues. Ionizing radiation to blood cells can cause hematologic depression and depletion of immature blood cells. Ionizing radiation adversely affects blood cells by depressing the number of cells in the peripheral circulation. A whole body dose of 0.25 gray in tissue delivered within a few days produces a measurable hematologic depression. This does by far exceed the normal doses sustained by the working population of the radiation industry. Therefore, the use of blood count tests for the purpose of dosimetry is not valid. Most blood cells are manufactured in bone marrow. Radiation causes a decrease in the number of immature blood cells or stem cells produced in the bone marrow, and hence a reduction ultimately in the number of mature blood cells in the bloodstream. The higher the radiation dose received by the bone marrow, the greater will be the severity of the resulting cell depletion. If the bone marrow cells have not been destroyed by exposure to ionizing radiation, they can repopulate after a period of recovery. Whole body doses of ionizing radiation can cause severe damage to the human body. Human beings who receive whole body doses in excess of 5 gray in tissue may die within 30 to 60 days because of effects related to initial depletion of the stem cells of the hematopoietic system. Damage can be reduced with the use of antibiotics and isolation. The term lethal dose, or LD, is used to illustrate the dose that produces death in 50% of the subjects within so many days. The common time frame used is 30 days for animals and 60 days for humans. LD 50-30 is used for animals, and LD 50-60 is used for humans because a human's recovery is slower than that of laboratory animals, and death may still occur at a later time after a substantial whole body exposure. Whether survival lasts for 30 days or 60 days, the lethal whole body dose for humans is generally estimated to be 3 to 4 gray in tissue without treatment, and higher if medical intervention is available. As previously stated, ionizing radiation affects primarily stem cells. Radiosensitivity is solely based on the lifespan of the blood cells. Lymphocytes and neutrophils are the most radiosensitive blood cells. They are white blood cells which help fight infection. A reduction in the number of lymphocytes and neutrophils mean a reduced ability to fight disease. Granulocytes are also a type of white blood cell that are essential in fighting infections. When exposed to ionizing radiation, granulocytes increase in number and then decrease greatly in number. Thrombocytes or platelets initiate blood clotting and prevent hemorrhage. Exposure to ionizing radiation lessens the number of platelets circulating in the bloodstream. When numbers are significantly depleted, it can take 2 months to regain its original numbers. Erythrocytes are less sensitive to ionizing radiation than any other blood cell. The normal white blood cell count is 5,000 to 10,000 millimeters cubed. Epithelial tissue lines and covers body tissue. It is highly radiosensitive because of its high reproductive rate. Muscle tissue contains fibers that affect movement of an organ or part of the body. Since muscle tissue cells are highly specialized and do not divide, they are relatively insensitive to radiation. Nervous tissue is found in the brain and spinal cord. Nerve cells in adults are highly specialized and do not divide, and therefore are relatively insensitive to radiation. However, nerve cells in an embryo are dividing rapidly and are very sensitive to ionizing radiation. Exposure to nervous tissue of an embryo can result in microcephaly, central nervous system anomalies, and even mental retardation. A safe dose of ionizing radiation to male and female gonads is 0. A radiation dose of 2 gray in tissue may cause temporary sterility for as long as 12 months, and a dose of 5 or 6 gray in tissue can cause permanent sterility in men. Immature spermatogonia are very radiosensitive. Exposure to ionizing radiation also may cause female sterility. The ova of a female fetus and those of a young child are very radiosensitive due to the immature cells. Women between the ages of 20 and 30 years exhibit the lowest level of sensitivity. After a woman reaches 30 years, the overall sensitivity of the ovaries increases constantly until menopause. Temporary sterility usually results from a single radiation dose of 2 gray tissue to the ovaries. A single dose of 5 gray in tissue generally causes permanent sterility in mature women. Think about why we ask women LMP in pregnancy. We have an ethical and professional responsibility to ensure the safety of our patients. Radiation protection is so important. Please review the summary section in your textbook to review all of the information we discussed. If you have any questions, be sure to email me or place on the discussion board so everyone can see our dialogue.